Welcome to the BioInsights, where science comes alive.
Today, we're diving into what is basically a high-stakes medical detective story, the complex biochemistry of protein estimation. Imagine a laboratory as this high-tech forensic lab, right? And the lab technicians, they're the detectives, meticulously working to uncover these hidden molecular clues floating around in our blood at this very moment. It's this incredible, completely invisible world, and the techniques we use to pull these secrets into the light, well, they're just absolutely fascinating.
Okay, let's dive into this. How exactly do doctors count invisible molecules? Whether a doctor is desperately trying to diagnose a critical disease in a patient, or maybe a researcher is running a complex SDS-page experiment to isolate a single enzyme, a huge question always comes up. How do you accurately count molecules that you literally cannot even see? Finding the exact concentration of proteins in a sample isn't just some abstract academic exercise. As you'll see, it is literally a matter of life or death.
To really grasp why this is absolutely crucial, let's look at the heavy lifters in your bloodstream, your plasma proteins. Now, a healthy human has a normal total protein baseline of about 6.5 to 8.5 grams per deciliter, and that mass is made up of absolute powerhouses. You've got albumin, which brilliantly maintains your blood's osmotic pressure so, your veins don't just leak. Then there are globulins, acting as your body's frontline defense and transport system. And of course, fibrinogen, which is the absolute key to blood coagulation.
So here is our roadmap for the explainer today. We'll be looking at why measure proteins, the magic of spectrophotometry, the UV absorption method, colorimetric assays, cracking the standard curve, and finally diagnosing human disease.
Section 2. The magic of spectrophotometry. Now, every good detective story needs an interrogation room, right? Well, in biochemistry, that interrogation room is the spectrophotometer. The core concept here is elegantly simple. Since we obviously can't count the proteins manually, we use light. Imagine you shoot a perfectly known amount of light particles, let's just say exactly 100 particles, at a liquid sample. On the other side of that liquid is a detector plate. If only 60 light particles hit the detector, well, we know the sample absorbed exactly 40 particles. By measuring the light that didn't make it through, we indirectly measure exactly how much protein is hiding in that tube. Pretty clever.
To do this accurately, lab technicians actually live by a three-step mantra: blank, standard, test. First up, the blank. This is usually just distilled water, and it's used to prove that 100% of the light passes through, basically zeroing out any machine error. Second is the standard. This is a solution where we already know the exact protein concentration, say 6 grams per deciliter, so we can see exactly how much light that known reference amount absorbs. And finally, the test. This is the big one, the patient's actual unknown blood sample. Let's move to Section 3 and see how this builds. The UV absorption method. This method relies entirely on a magic number, 280 nanometers. This is the precise ultraviolet wavelength where aromatic amino acids within proteins, specifically tyrosine, tryptophan, and cysteine, just naturally absorb light. By shining UV light at exactly 280 nanometers, boom, we get an instant read. Now, if there's any stray DNA getting in the way and muddying the waters, scientists use a really clever mathematical tool called the Warburg-Christian formula. That lets them correct the reading and find the true concentration. Now, the UV method is a massive favorite in the lab because it's incredibly fast. Using a neat little device called a nanodrop, scientists literally only need a tiny 2 microliter droplet of sample to get an instant reading. But, it's not perfect. It can be easily messed up by the presence of DNA or even just standard laboratory detergents. So to get around that sensitivity, scientists often turn to our next technique, colorimetric assays. Now, these do take a little longer and require adding extra reagents, but they are highly specific.
Section 4. Colorimetric assays.
And this brilliantly illustrates the difference. If the UV method is direct observation, these assays are kind of the chemical disguise approach. We add specific chemical reagents to the invisible protein samples, and they literally change color right before our eyes. It's like magic. The intensity of that brand new color tells us exactly how much protein is in there. Let's look a little closer at how a few of these actually work in practice. First up, the burette method. This is basically the gold standard for measuring total proteins. We place the serum in an alkaline medium and just unleash copper ions into the mix. It is absolutely amazing to see how these copper ions actively hunt down and bind to sequences of 5 to 6 peptide bonds in the proteins. This creates a really distinct violet-purple complex that we then read in the spectrometer at 540 nanometers. Oh, and we also add tartrate to stabilize the reaction, plus iodine to prevent the copper from, just reducing itself automatically. Then we have the Bradford assay.
Now this one relies on an unstable cationic dye called Coomassie Brilliant Blue G250. In its free unbound state, it actually has a reddish-brown color, peaking at 465 nanometers. But when it binds to a protein, literally anchoring itself to specific amino acids using electrostatic and van der Waals forces, it completely stabilizes and shifts to this vibrant, highly readable blue at 595 nanometers.
But what if we don't want to measure total proteins? What if we only want albumin? Well, for that, we use the BCG, or bromocressyl green method. In an acidic pH, this anionic dye specifically targets albumin, shifting from a yellow-green to a green-blue at 620 nanometers. This is so vital because albumin makes up over half of your serum proteins. To put that into perspective, in a healthy person, the ratio of albumin to globulin is typically 2:1. If that ratio flips, well, doctors know for sure they have a major problem on their hands.
Section 5. Cracking the standard curve.
So how exactly do we translate those beautiful lab colors into hard, actionable diagnostic numbers? Well, technicians create a sort of lineup of suspects. They take bovine serum albumin, or BSA, and create serial dilutions. Basically, these are samples with increasingly exact concentrations, stepping up from 0 all the way to 1,500 micrograms per milliliter. They add the color reagent, read the exact light absorbance for each individual sample, and then plot those points on a graph to draw a straight, predictable line.
This line acts as our ultimate translation dictionary. When we test a patient's unknown sample, we just measure its light absorbance, find that exact spot on our newly drawn curve, and trace it directly over to the exact protein concentration. By basically calculating the absorbance of the test divided by the absorbance of the standard, multiplied by the standard's known concentration, the code is completely cracked.
So the crucial point is, Section 6, Diagnosing Human Disease.
Why do we go through all this abstract math? Because at the end of the day, these numbers represent human lives. When the standard curve spits out a number drastically below that normal 6.5 to 8.5 range, we diagnose hypoproteinemia. This gives doctors massive, life-saving clues. It might mean the patient simply isn't eating enough protein, like we see in severe malnutrition cases like kwashiorkor. It could mean a failing, cirrhotic liver has entirely stopped synthesizing proteins. Or it could even mean that kidneys are damaged, like in nephrotic syndrome, and they're accidentally excreting the body's precious proteins directly into the urine.
Conversely, what if the numbers are dangerously high? That is hyperproteinemia. Now, this can happen through a sort of concentrational illusion, like in cases of severe dehydration. Because there's less water in the blood, the proteins just seem packed tighter together. But it can also indicate a true, absolute spike in proteins due to chronic infections, where the immune system is just churning out acute phase proteins. Or, more ominously, it could point to a plasma cell disorder.
Let's look at a real-world medical mystery from our sources to put this all together. A 65-year-old male comes into the clinic. He's experiencing persistent, deep bone pain in his lower back and his ribs. He's fatigued, he's losing weight. The doctor runs a serum total protein test using that burette method we talked about. The results come back showing massive hyperproteinemia, and a urine test reveals something called Bence-Jones proteins. So as our medical detective today, what do you think is destroying this patient's bones?
Now, what's really interesting about this slide is the answer. It's multiple myeloma. It's a cancer of the B lymphocyte plasma cells. Basically, the cancer causes these cells to uncontrollably overproduce gamma-globulin antibodies. Those crazy high total protein numbers on the spectrophotometer, yeah, that was the cancer. And those Bence-Jones proteins in the urine? Those are the specific cancerous antibodies being desperately filtered out by the struggling kidneys. The lab test essentially caught the killer red-handed.
It is profoundly amazing, isn't it? The fact that a simple drop of copper or Coomassie dye in a tiny glass tube, placed into a beam of light, can literally save a patient's life by revealing what is totally invisible to the naked eye. From calculating standard curves to diagnosing bone-destroying cancers, protein estimation really is the bedrock of clinical biochemistry. So as you go about your day, it really makes you wonder, what invisible chemical clues are flowing through your veins right now, just waiting for a simple color change to reveal their deepest secrets?